Okay, I'm James Tucker. I'm in the Department of Plant Science. I'm doing my PhD and working in biotechnology approaches for Fusarium head blight breeding in barley. This picture is a 6-row barley spike, Hordeum vulgare, and this is some pictures of the domestication of barley. Barley is an ancient grain. It was domesticated about 8,000 years ago. And this was done by early agrarians where they selected genotypes and they were bred for the threshability. Barley is used mostly for animal feed, but a select portion is selected and used to produce beer. Other usage is through food, and there's a lot of health properties associated with that. So sustainable ecosystems are based on pillars. Environmental health is important. The crops that are produced have to be economically profitable, and there should be social and economic equality. So these all have to work together. There are several agroecological properties that barley conveys. It has a low nutrient and water requirement. It's strongly weed competitive. It produces organic material that it's worked back into the soils, and it's able to be grown on marginal lands. So I work in a barley breeding program, and barley breeding increases profitability through genetic enhancement. So this is possible through conferring genes for protection against plant diseases and insect pests. This is a sad graph about barley production in Canada. There's been a steep decline of production in the last decade. This is likely due to profitability and competition with other more profitable crops. One of the reasons why barley is no longer profitable is due to this fungus, Fusarium graminearum. So this depicts the life cycle. This was introduced a long time ago from England, but it became a problem in barley in Western Canada in the 1990s. Here's some visual of the disease of a 6-row spike, and that's on the left is a 2-row spike. These are the fruiting bodies that the fungus produces on the residuals that are left in the field, which turn into spores and infect the heads. One of the main problems with this disease is it produces mycotoxins or secondary metabolites that the fungus uses to invade the plant or to protect itself in its environment. So these are toxic to humans, and we don't want that in our food chain. There are several ways plant breeders can breed resistance. There are genes that confer qualitative resistance or major genes, and the disease resistance I'm working on is quantitative. This is a more difficult disease to breed for. Here's some pictures of the Fusarium head blight nursery in Brandon. We culture the fungus, produce inoculum, and we spread that in the field under irrigation. We make ratings, and it's a laborious process because all the plots have to be harvested for the mycotoxin assay. I'm working in my PhD on developing genome-wide association studies to look at gene markers that cover the whole genome, which is more applicable to quantitative resistance. Here's some photos of DNA extraction, so sampling in the field. So take bits of the plant tissue, the cells are lysed, and the DNA is removed from the plant tissues. It's concentrated, and these can be used then for further assays. So one of the methods I'm using is using an aluminum eye scan, which is microbead technology where many small beads are fixed with DNA probes. These detect SNP polymorphisms or single nucleotide polymorphisms. This is a picture of my association mapping. To do this, we use kinship relationships to remove patterns that are from the sisters and of the lines, and these graphs show how we look and find significant genes. Another approach I'm using is genomic selection. So breeding takes a long time. It can take about 10 years to produce a variety. We can use genetic information of whole genome markers using 40,000 markers to make selections. So this is a picture of another technology I'm using. This is next generation sequencing, where genomic libraries are made and they are sequenced on a Illumina instrument. So you can get lots of information out of this, and this is aligned to genomes. My experiment that I'm using is using doubled haploid technology. This is in plants. I'm looking at a doubled haploid variety that was produced with in vitro selection from a parent malt line. So I'm comparing that parent line to the line that was developed. And I'm conducting an RNA sequencing experiment where looking at the gene expression of the parent line versus the selected line. So hopefully I can identify genes that are associated with resistance to Fusarium head blight through that method. I'd like to thank you for your attention.